Thank you for that very kind and warm introduction. So pleased to be here to talk about ideas underlying the Next Gen SIBS project and understanding autism risk in grandchildren. So as we're all aware, with the ongoing rise in prevalence in ASD that's now been going on for at least 2 decades, there are ongoing concerns about risk. And for parents of children in that original generation, the concern is particularly pronounced for the new generation of offspring, these grandchildren. And we don't know a lot about what the exact recurrent risk could be. So it is a big, important question, both from the standpoint of public health, as well as understanding even the underlying biology that goes into contributing to that risk. And given that prior family studies have been so instrumental in understanding the heritability of ASD in general and establishing that, I think it's fair to say that studies of these new generations now have the opportunity to play a critical role in estimating recurrence risk, which in turn informs biology, as well as provokes opportunities to improve clinical practice. So as I mentioned, ASD is a heritable condition. This is well established. Twin and family studies have played a big role in clarifying this, with upper limit estimates of 80 to 90% of the variation in ASD being due to genetic factors. And molecular genetic studies have bolstered these findings in terms of identifying numerous genes that have also been linked to the causation of ASD. And so the convergence between these two lines of work has helped us to understand that it appears right now that common additive variation, variation that adds incremental risk to the likelihood of developing ASD, appears to be at play here, and that the heritable factors that underlie this risk apply both to people who have a diagnosis of ASD, as well as family members who have the risk but not the diagnosis, and even in the general population as well. And so family studies then are in this very important position of being able to provide understanding of like a broader variation across the population so that we can better clarify the multitude of genetic factors involved, as well as having the measurement capability of sort of that titration of risk that leads us to understand the distributions of autistic traits and liability better in the population. And in addition, one other point I'll make that speaks to early life, we do have behavioral markers that in some cases are relatively accessible or feasible to do clinically, parent report instruments of autistic traits that can kind of give us an index or a sort of proxy measure of heritable liability. So this histogram here comes from work in a general population twin study that I worked on with John Constantino, and we basically were able to show that autistic traits can be measured in a continuous distribution across the population at 18 months, and they are highly heritable. And then this picture should look a little familiar as well, showing that a more direct behavioral measure involving eye tracking that measures something called social visual engagement, which has been found to be linked to ASD risk, is also something that's measurable very young. And by age 18 months, we're able to tell that that also operates under heritable influence as well. So again, this puts us in a position where understanding what's going on in the new generation, how ASD is emerging, and what behaviors we can track to measure that can definitely inform our understanding of biology and recurrence. But not only that, there's also an important public health concern here as well. So as I mentioned, families are very keen and interested, understandably, in knowing how best to serve their children and to put them on the appropriate path, that maximizes their outcomes. And so a well-coordinated family study that brings researchers and clinicians together in a way where risk assessment can be refined, which then can lead to more timely early intervention to produce or to promote outcomes, as well as opportunities to provide family support. And with multiple generations in a family, this support doesn't just have to be to that new generation, it can also be to the older generations that we're still needing to learn so much about. So work with family studies across generations kind of puts us at this sort of unique intersection where there's a position or an opportunity rather to understand biology and improve clinical practice. Now, one of the areas of biology that we can kind of pick up on by observing the phenomenology that sort of comes together with the epidemiology of ASD has been the sex ratio. So this is something that has been observed relatively consistently in different cohorts across time, and even different cohorts that have sort of different diagnostic sensibilities. And so relatively consistently, there's a ratio somewhere of about 3 to 4 to 1 males to females. There has understandably been a growing movement acknowledging that perhaps females are underdiagnosed. This is an important line of research as well. However, as best as we can tell, in infant sibling populations where you have individuals who are at high risk, high familial risk of ASD, who are studied in a very rigorous manner from early in life, we still see that sex bias towards males. So it certainly appears that at some level, this sex bias is reflecting some disparity related to underlying biology. And so one issue with that then that would be a logical hypothesis would be, well, maybe there's some sex linkage going on here that accounts for this phenomenon of epidemiology in the population. However, we know that most of the risk in ASD has been found to be located on autosomes genetically. So this kind of rules out that possibility. And in addition, there's been more and more work looking at a variety of populations suggesting that females appear to have a higher liability threshold. So this is somewhat the idea of female protective effects. This has been supported by findings that in some cohorts there's a larger mutational burden for genetic liability for females who ultimately get a diagnosis. In cases where highly deleterious mutations are responsible for ASD, we find that in those cases the male and female disparity is sort of attenuated or lessened, implying that heavy hitter sort of puts the females more on the same playing field as males. And in families that are enriched for females who are affected with ASD, there's some studies that have shown evidence that there's a greater rate of recurrence in the other family members for those female cohorts, suggesting that there's more liability traveling in those families. One way to illustrate this type of phenomenon is through a pedigree that was observed actually through some of the more molecular genetic findings. And in this case, it was related to SHANK1, a gene that's responsible for a protein that regulates transmission between neurons. And so with these pedigrees, hopefully it doesn't look too confusing, but each row of lines is going to represent your different generations. So again, an opportunity to see how understanding transmission across different generations can help us understand ASD biology. Squares are males, circles are females. Dark colors indicates an ASD diagnosis, and where you see that 63.8 language, that indicates that there's a deletion in SHANK1, which has been linked here as a causal factor for ASD, but only in the males. So you should see both transparent-looking circles as well as the dark squares that have this SHANK mutation, and it looks like only the males are the ones who are affected. So here again is an example showing that by examining how ASD travels within families, we can get a feeling for these sex disparities. We can get a feeling of where there's something surprising relative to what the underlying genetics seem to say is there as a risk, and what the different sexes of the individuals show us about how that risk is expressed. So this in turn then can lead to some sort of model where there is this idea of core underlying heritable ASD liability. And so how would we explain that sex maybe appears to modulate the expression of this liability? And so one way to think of it would be that in females, perhaps there are some protective factors, some compensatory factors, heritable that essentially decrease the expression of that ASD liability, making it look less. Another alternative explanation would be that in males, there are susceptibility factors that almost amplify the effects of that liability, so that now what would not lead to ASD in a female is more likely to lead to ASD in a male. And these distinctions are important to keep in mind for two reasons. So one, from the standpoint of genetic studies, it can help to know what types of factors to look for in which sex. So am I looking for protective factors in females, or am I looking for susceptibility factors in males when I'm doing behavioral genetic studies, for example? Another reason is also going back to that public health concern. If females have more masked liability, then what that implies is that the recurrence rate in the next generation may be much higher than expected. And so that's going to have impact on the population in general. So these types of questions led to the postdoctoral fellowship that I was fortunate enough to get funded by ASF, the second generation survey project. John Constantino was my mentor on this project, and to try to get a handle on these questions, we wanted to collect a three-generation cohort, starting with grandparents who would report on the incidence of ASD in their offspring, and these grandparents were required to have a child with ASD. As well as their grandchildren. And we chose grandparents because they often are very sensitive to these types of concerns, sometimes more so than busy parents, and also because they could provide information on a larger number of members of the family cohort. And through this type of work, then, we are able to ask questions about whether unaffected sisters of ASD probands have higher rates of ASD in their offspring that would be expected in the general population, and ultimately, have the possibility to compare that with the unaffected male siblings to distinguish between the different types of mechanisms that might underlie sex modulation. We worked with the Interactive Autism Network, Telethon Kids Institute to get our cohort together. We used a web interface to allow families easy way to get count data. And we also went ahead and collected survey information to provide some behavioral phenotyping. And in the future, actually, this collection is still ongoing with the help of our collaborator, Laura Klinger at UNC Teach. And so these are the results that we have found preliminarily. So one thing I should point out, this was and has been a challenging cohort to collect. It's been 3 years and we've gotten 150 families. Here, what I've focused on is the offspring of the unaffected full female siblings. So that's 88 full female siblings. It's from 75 families. We had grandparents report both on ASD in that grandchildren generation as well as suspected ASD, because there's been evidence that grandparents can sometimes have a good pulse on that, and that also there's a correlation between those two things. And this kind of provides us a range in terms of understanding the upper limit of what the recurrence rate might be. So the top few rows pulled, or 2 rows, pull all the both males and females together. And so if we think of a general population prevalence rate of somewhere, 2% would be a high estimate of that. One can see whether you take ASD diagnosis or suspected ASD diagnosis with that. Certainly in these unaffected female siblings, there is an amplification of risk pretty substantially. And that's also true when you separate by the male and the female grandchildren as well. And so for us, what this meant is that there's certainly a cause for concern regarding the silent transmission through the females. We don't have as many unaffected male siblings in our cohort, although I will say at this point, the female recurrence rates are higher in that regard, which would be similar to what we would have anticipated from a female protective effect type hypothesis. Given that there are lower numbers than we had hoped to get with that prospective approach, we also have gone ahead and reached out to a collaborator, Sven Svanden, who has access to data from the Swedish National Register, which can also be linked up with a multi-generation register. So what this table outlines here is the number of participants in each of those generations. It includes a parental generation and an offspring generation here. And in this way, with this broad general population population registry, relatively equitable health access, common ways of assessing diagnosis, and the ability to link diagnoses across different generations, we can kind of recapitulate some of the features of the prospective cohort and go ahead and try to ask questions about counts of ASD in the latest generation. We've just started this analysis. At this point, we know that offspring generation has levels of risk that seem comparable to what's expected by today's standards. So hopefully this kind of introduction into the type of the work that we've been doing helps make the case that family studies that include multiple generations can provide us insight into the biology underlying ASD, ways to help families by engaging them in the research, and also very practical risk estimates for the future. I do think that there's great hope that this type of method and approach ultimately will allow for a more personalized risk evaluation. So there are methods. We know more and more about the underlying genetics today. If families were enlisted in sort of long-term studies, there'd be opportunities to, for example, genotype probands to see if that tells us something about the genetic architecture and risk in a family. Study behaviors that would allow us to have subtyping that lets us add to that information to provide a better risk estimate. And hopefully identify neural signatures that might be able to also sort of combine with these other aspects of measurement to refine the risk measures. And then, of course, ultimately inform mechanism and lead to more individualized interventions. So I'd like to acknowledge Dr. Constantino, who again has helped mentor me through all the beginnings of this, and as well as our collaborators over at IAN, TKI, Telethon Kids Institute, the Swedish National Register, our upcoming work with Teach and Laura Klinger, and our funders without whom this wouldn't have been possible, the Autism Science Foundation, as well as NIH, NICHD, who provided John Constantino with funding to continue this through WashU IDDRC. Thank you.